Well, hello to everyone, and thank you for logging on for our final installment of our 2021 virtual horse management seminar series. I do see that we still have people coming in, but we do have a full program tonight, so I want to go ahead and get us started. I am going to introduce my advisor during my PhD program here at Rutgers, who's Dr. Carrie Williams. Dr. Williams earned her doctorate degree with an emphasis on equine nutrition and exercise physiology in June 2003 from Virginia Polytechnic Institute and State University. She holds a master's degree in equine nutrition, also from Virginia Tech, and a bachelor's degree in equine science from Colorado State University. She joined Rutgers University in July 2003 as its equine extension specialist and associate director of outreach for the Equine Science Center, taking an active role in teaching, conducting research, and working with the equine and academic communities to ensure the viability of the horse industry in New Jersey. Thank you, Dr. Williams.
Thank you, Jen, Ellen, and Kyle. They have all helped me tremendously put this together, and I really appreciate all the help. And I really appreciate everybody for joining in. Today, as Jen said, it is our last in the series of our horse management virtual horse management seminar. So thank you very much. I'm really excited today because, as Jen mentioned, I am an equine nutritionist. So I'd like to say I saved the best topic for last, but then again, I am a little partial to that. So before we get started, I want to do just a couple quick fun things, I guess. I think it's fun anyway. So everybody out there is going to need to be able to log on to another web browser. So you could have your phone, you could put it in your phone. You could close out this browser, open up another one. You could have an iPad next to you, whatever you want to do. Just do me a favor and log on to menti.com. Let me type that in the chat here. It's MENTI.com. It's pretty easy. So I want everybody to do that. You're going to just see a screen and it's going to have a login number. I'm going to give you that number in just a minute. I'm going to do 2 questions for you. And we're going to see where everybody's from and what you guys do. We've kind of made it our goal to reach all 50 states in the US. I don't know if we're going to do it today, but we're pretty darn close. So I really need to know what everybody's state is. And if you would do for me a favor, when you do type your state, don't abbreviate it, spell out the whole name, and you'll actually see why. I know, unfortunately, if you're from Massachusetts or Tennessee, it might be a little bit long, but please spell it all out because there'll be a reason why. And we're also going to go through, not yet, don't put it in the chat yet. We're going to do it in Menti. I just haven't opened Menti yet, so you can't do anything yet. And I want to know a country. So you don't have to put USA. If you're in the USA, put your state. But we've been having a lot of people from other countries. I think so far what I've seen Brazil and Peru and Australia and Mexico, Canada. I'm probably missing some, but we've seen a lot. So I really want to see where you guys are logging in tonight. So if I gave you enough time, menti.com, I'm going to go ahead and share my screen. And then you're going to type in this code at the top. So it's 6951530. So if one of my techie helps can actually go on there and help me type in the chat so you all see it, 6951530, and type your state or your country. And you see, this is a fun little word cloud. And as everybody's logging on, we got 36, 39, 41. This is the number of people logging on. You see the word cloud change. The bigger the word, the more people have entered that word. So far it looks like New Jersey's on top, but Pennsylvania and Minnesota are close behind, which makes sense again, because both of our speakers tonight are from Pennsylvania and Minnesota. So they probably have their tried and true followers. Now let's see, where else do we have? We've got next up coming in at West Virginia, North Carolina, Wisconsin, New York. It's kind of hard to read the words as everything changes and scatters around the screen. But where are some other places? We've got Arizona and Georgia, Montana. What about some countries? Do we got any EU countries out there? I see Canada and Mexico. Peru's back. That's good. Awesome. Brazil, Mexico. I think I already mentioned Mexico, Canada. So I'm going to leave it open for just a little bit. We've got 87 people that have actually logged into Menti. If you're just starting now, logging on to the webinar. We're just finding out where everybody's from. So go ahead and log on your phone or on your computer or iPad or wherever, menti.com. And then you type in the code 6951530 and type in your state or country. Like I said, we've got, we have Alaska back. I know we had Alaska and we had Hawaii in one of them as well. I don't see Hawaii back this time around. So I'm just going to leave it open for a little bit. I don't want to take too long away from the speakers, but I always think it's fun to see this all change and move. But we seem to have the same themes. Obviously, New Jersey comes in at the top. I think it has been for the last couple. Minnesota and Pennsylvania close behind, which I think have been for the rest of them as well. I see British Columbia in there. Hello, hello. Awesome. All right, well, I'm going to do one more slide and I'm going to find out now what everybody does for a living, or with horses in general. So the next question is, what is your primary interaction with horses? So are you a horse owner? If that's pretty much what you do with horses, then that's great. I know that's what I am. Lessons or trainer, a barn manager, you veterinarian, an industry professional, horse enthusiast. So you may love horses, but not actually own one at the time, or anything other. So other can be, I know I have a picture of a farrier here, but dentistry, equine assisted therapies. Are you a student? We could probably list a bunch of these. If you want to tell us what you mean, there's 20 of you that said other, type in the chat what you mean by other. What do you do out there? So go ahead and chat to us and let us know what you do. So we've got 77 people answering this question. I know we have a few more online. It looks like we have 144 total attendees. So we still have a couple people we're waiting on, but I'm not going to give it too long. We have at least one veterinarian, a couple trainers, a couple barn managers. But same as what our past couple webinars have been, there's been a lot of horse owners and a lot of other things. So that's great. We've got 81 people that logged on. Well, thank you again. I'm going to just stop sharing so that we can introduce our first speaker and then he can start sharing his slides. But thank you all. I really think it's fun to kind of see where everybody is from and what everybody is doing. So thank you guys again. It looks like an other quickly. Oh, we got a barrel racer. That's fun. A groom. I've seen that one a couple times. Therapeutic riding, lessons, a student. Great. Another groom. Awesome. So we got a lot of fun things. All right, so without further ado, let me go ahead and introduce our first speaker, Dr. Burt Staniar. He is currently at Penn State. He joined them on their faculty in 2007. However, I want to deviate from my script and just a wee bit, just a little bit, because I don't know if how many of you know Dr. Staniar, but you heard not Dr. Whiter yet, but Jen read my bio. I did my graduate work at Virginia Tech. Well, so did Dr. Staniar. We overlapped. I would say I was kind of a lowly little master's student as Dr. Staniar was doing his doctorate work. And he helped me answer a lot of my questions. I did a lot of glycemic, insulinemic work, looking at different diets. And that was a lot of what Dr. Staniar was doing at the time and looking at cartilage and skeletal development in thoroughbred horses as well. So when he finished up his doctorate at Virginia Tech, he stayed on to do his postdoc. And then after that went to to Penn State, where he is now. He's done a lot of research that has shifted to gastrointestinal health, assessing the luminal environment within the GI tract and gut permeability. He teaches a lot of courses, and I will say he's been helping my graduate student, Jen, out a lot with looking at particle size, different fiber sizes of different diets, which I could think of no one better than to give this talk about how important fiber is to the horse's gut than Dr. Staniar. So I am now going to shut up and turn it over to Dr. Staniar. Take it away.
Dr. Williams, thank you very much. I'll go ahead and share my slides here. So first of all, I want to say thank you very much to Rutgers for having me in. It's really always a pleasure to speak to this group. Rutgers has an amazing equine program. They do an incredible job bringing together some good speakers. And it's kind of an honor to really be invited to do this talk and to talk to this group of individuals. Clearly, Dr. Williams and her team have done a wonderful job bringing together a large group here. It looks like right now, I'm just sort of looking at the numbers. We have about 148 people on. So that's absolutely phenomenal and really exciting. I've also really enjoyed, it's funny, Dr. Williams said that we were working on some of this particle size stuff together. It's really great, having worked with Jen on some of this, and I would say that she's doing the majority of the work and really helping me along with some ideas that I have developed over the years. And I think that this area of fiber and dietary fiber for horses is really, really exciting. Finally, I'll say that it's awesome that I get to do this with Dr. Martinson, who you guys will see afterwards. I think our talks will complement each other really well. And so that's going to be a lot of fun. My talk is going to be a little bit sort of, we're going to develop some ideas here. And I look forward to seeing what some of the participants put into the question and answer and the roundtable we get to have at the end of all of this, because I think that there is such a huge and exciting future in the area of equine nutrition. And, it may be amazing to many of you, you think of the long history that humans have had with horses, to think about the future. And to think about the fact that there is still a lot that we can do to ultimately improve the well-being and the performance of the horses that I think are so important to all of our lives. I mean, I grew up around horses. I've been riding since I was about 4 or 5 years old. They've always been a part of my life, and I'm thankful that they are. So that's really exciting. Anyway, today we're talking about fiber, and I really, I've been talking about dietary fiber for horses for quite some time, developing some ideas here. Really much of this came, and it's funny how ideas develop over a long period of time as they're kind of simmering in your head. But this slide really brings together a lot of things that I'm kind of thinking about that I think are kind of interesting in relation to fiber. At the center of the slide, you'll see a picture of fire. And my academic grandfather is this guy named Max Kleiber. And he wrote a really important animal nutrition book called The Fire of Life, which talks about nutrition and the fact that nutrition feeds the energy of all animals, and studying that nutrition and trying to understand that. And I heat my house with wood, and I love fires. But the fact of the matter is, when you look at that wood that's burning right here, much of that wood is made up of fiber, lignin that we're going to talk about, all coming from plant material, right? And if you look on the left-hand side of the slide, of course, we see the horses and their fire and their life comes from much of the fiber that they're eating. This is really the diet that they have adapted to eat over millions of years. And it's interesting to note further that we continue to use fiber. And this gentleman here actually studies the utilization of grasses as potentially greener sources of fuel. So thinking about, again, actually burning grasses, and some of you will be familiar with this as we think about other fuels that we might utilize into the future. So it's really fascinating to make the connection that we see with fiber and its source as a fuel for both living animals, but also that we see as a source of fuel for many other things. So, in a big context of things, I think it's really interesting to think of things in that bigger picture. Fiber for horses. Yeah, I think everybody that's probably in the presentation today listening to this, they all know, I think, and that most horse owners understand that fiber is important for horses. They all know that their horses eat grass. They all know that fiber does supply some important things for the horse, but I'm not sure that everyone really understands some of the details of it. And as one of the things that I hope is that at the end of this presentation, that you all have a little bit clearer picture of exactly what fiber is. You can define that a little bit more clearly. You can talk about the individual components of fiber and think about how you might modify the fiber in the diets of the horses that you're either feeding or helping your clients out with the way that they're feeding their horses to make a little bit more precise adjustments to some of that fiber that might benefit, again, my big idea is the well-being, the health, but also the performance of those animals. The two examples of fiber that I have on the slide here at the top is psyllium husks, which many of us from a human nutrition understanding might think of as Metamucil. And that's an important and often used soluble fiber source for human beings. And in the lower part of this picture, you'll see beet pulp. Again, a fiber source that many equine nutritionists will be very familiar with. And I love beet pulp. I think it has a lot of potential benefits to horses being fed in a range of different ways. So we'll try to understand all of that. Dr. Williams mentioned the fact that she and I have done a lot of research together over the years. The equine scientific community is a very close one, and we all work with one another. We all know one another. She and I have done a lot of work together in the area of glycemic and insulinemic responses, sugar and starch diets. Again, I think this is something that many of you will be so familiar with, right? We have all these diets that they talk about the amount of starch that are in those diets. So they talk about whether they're high insulinemic or low insulinemic and how that influences the horse's glucose response. We worry about things like insulin resistance and metabolic syndrome, to the point where I think it becomes very confusing. We have all these different terms, and yet one of the other things that I want you to think about is carbohydrates. So horses eat plants, and plants have a lot of carbohydrates in them. So there are two big categories of carbohydrates that oftentimes, again, if you're any student of mine that's taken an equine nutrition course with me or done anything, I always tell them, if somebody says carbohydrates, you should immediately think about non-structural carbohydrates and structural carbohydrates. And as soon as you think about non-structural carbohydrates and structural carbohydrates, well, non-structural, you think about sugar and starch. Those are non-structural carbohydrates. And when you say structural carbohydrates, well, immediately fiber should come to mind. That's a structural carbohydrate. And we'll talk about some examples of those, things like cellulose, hemicellulose. Lignin is not a carbohydrate, but we will talk about that some. So, there's a lot of terms that you have to become familiar with as an equine nutritionist and as a horse owner. And the more you read about those and hear about them, the more useful those particular terms become for you. So the horse eats grass. Grass is an absolutely amazing plant, as are many of the plants that are an important part of our horse's diet. And there are some relatively challenging environments that those grasses can grow in. This is a picture of the Gobi Desert in sort of the Mongolian region of our planet, and an area where we have a very harsh environment, yet a place where, look at the extent of grasses that are growing there. And this is an environment that the horse is very well adapted to. This is an environment that the horse evolved in over millions of years to make use of that extensive, abundant source of fiber and other types of carbohydrates. There's so much of that plant material that's out there. Now, it's not always available. Okay, so there are times of year where that plant material becomes much more scarce. But it's really amazing to think about the plant that's growing there and the different types of forages that we see grow all around the world. It's great to see that we have some participants in the talk today from different parts of the world. So it's important to think about those grasses in different parts of the world and how they contribute to the horse's diet. So as we think about fiber, I really want you all, as we're going through my talk today, to begin to think about, well, how do we talk about fiber? How do we talk about that plant that I know my horse eats? I know that it's got hay in the stall. What are the important characteristics of that plant? And I'm going to take you in two different directions. One, I think we're very familiar with, and I am as a nutritionist, it's challenging to understand it, but that's the chemical characteristics. We're going to talk a lot about how we can look at the chemistry. Chemistry is very quantifiable. It's very objective. We can talk about exactly how much cellulose or hemicellulose or particular types of fiber are present there. What becomes a little bit more challenging is talking about some of the physical characteristics of that fiber. How does a plant necessarily change as it becomes more mature? And so as you look at the hay or the fresh forage that you're feeding, or maybe even the concentrated feed that comes from a feed company, how do you begin to think about the fiber that's there and characterizing that in a number of different ways? So I want you to be a little bit more familiar with the ways in which we can characterize that, thinking about how we can talk to one another, how can we communicate about that. So here's the first way. I showed this slide to my 12-year-old daughter, Molly, and we were sort of talking about things. And I said, what do you see here? Now, she's 12 years old. She hasn't done a lot of organic chemistry, right? But this slide let me get my little annotation tools here. I'm going to go ahead and get my spotlight so you guys can see what I'm talking about. On the left-hand side here, we're looking at glucose and fructose and sucrose. Those should all be sugars that you all have heard of. So those are the chemical formulas for those particular sugars, right? And then this top molecule here, this is starch. So right here at the top of this slide, we've got all of our non-structural carbohydrates. And interestingly, and of course, I've got all my little Zoom features here in the way, down at the bottom of this, we've actually got a cellulose molecule. Okay, so it's interesting looking at the comparison between starch and cellulose. They look very similar, but they're very different carbohydrates. One is a non-structural carbohydrate. The other one is a structural carbohydrate. The starch is inside the plant cells. This is a part of the plant cell walls. Now, on the right-hand side of this slide, I decided to detour a little bit. And I want you guys to keep this in mind as I'm going through the slides tonight. On the left, we've got carbohydrates. On the right, we've got hydrocarbons. Okay. And hydrocarbons should be something that you're also very familiar with that help you travel, just like your horse. Except on the right-hand side, this is how we get our horses to the various different events that we do, because we put them in the trailer and we pull them with our trucks. So at the top of this slide, we've got gasoline. These are some of the molecules that are present in gasoline and some of the hydrocarbons that are a part of gasoline and actually allow it to provide energy to our engines. And at the bottom of this slide, we've got a little bit longer hydrocarbon, and the longer hydrocarbons are actually molecules that make up diesel fuel. I find this amazing that these two sets of molecules, carbohydrates and hydrocarbons, are still the fuels that we use for travel, whether it be our horses or our trucks. And interestingly, all of these the carbohydrates and the hydrocarbons, you know what? They all originate from plants, right? You all know that. And that to me is amazing. These are these plants capturing all of this energy from the sun, depositing it on the earth in one form or another, and then being able to utilize it all as an energy source, whether it be for our horses or for our cars. And it's important, as I will point out to you, to understand all of these. I don't care whether you're feeding your horse or feeding your car. So we'll be thinking about that as we go forward. Right. Yes, I highlighted cellulose down here, but this is our fiber down here at the bottom. Okay, so that's what we're talking about. So, wow, just like everything else, carbohydrates can be very complicated, and we can talk about all different kinds of analyses of those carbohydrates. I've told you about structural and non-structural carbohydrates. On the right-hand side of this slide is a figure that, as an equine nutritionist, I love. This comes from Dr. Rhonda Hoffman in an excellent paper she put together years ago. Dr. Hoffman is now down at Middle Tennessee State. She's also a co-author on a new edition of The Horse that's come out, a book that I highly recommend. It's an excellent book. But Dr. Hoffman put this figure together, in an attempt to kind of differentiate all the different carbohydrates that horses eat. And tonight we're talking about fibers. So we're really looking at the lower half of this picture and all these different fibers that are here. And in the presentation tonight, I'm not going to begin to be able to really touch all the various different components of the fibrous material that horses eat, but we're going to talk about things like cellulose and hemicellulose and lignin. And we're going to talk about what ADF and NDF are. So you're going to understand those a little bit more. But ultimately, they are all parts of the plants that horses eat. And that's what's really important, okay, is that you have to understand plants a little bit more. And I would say one of the best things that horse professionals and owners can do is to become a little bit of an agronomist. Go out there and get a book on agronomy or get a book on forages. Learn about plants, because the more you learn about this feed stuff that we feed to horses, the better a horse owner you're going to be. So understanding some of these fractions is really important. And so you're going to see this slide again in the future, but we're going to talk about some of these chemical fractions. So again, here we see again that hemicellulose, cellulose, and lignin. These three pieces right here are the primary components, chemical components of the fiber fraction of this grass plant that's growing right here. Okay, inside the cells, you may actually have some sugars and starches that are actually inside those cells that the horse can use as an energy source, but they can also use all of these different sources of fiber, particularly the hemicellulose and cellulose as an energy source. And if we think about those from a feeding perspective, it really provides us infinite number of combinations of how we can mix and match all of these different carbohydrates with one another for horses that have different needs. All right, so let's talk about how we define some of that fiber from a chemical perspective. And of course, if you're a really famous scientist and you do really great work, you get various different things named after yourself, right? So we've got the Wend proximate analysis of feeds, and we've got the Van Soest proximate analysis of feeds. Well, we're going to move past the names at the moment. The Wend approximate analysis of feed is a much older approach, and many of you can look on the feed tags of the bags of feed that you have right now that you're feeding to your horse, and you can probably find how much crude fiber is there. This crude fiber is a measure of fiber. And no offense to anyone that's putting together the feed tags, but it's not a very good measure of fiber. It's very old. It's a very crude measure of the amount of fiber that's there. And it's not particularly useful to us from a nutritional perspective. Okay, it's one component of the carbohydrates that necessarily make up the feed. However, Van Soest, Dr. Van Soest made some significant progress here where we began to think about the plant and the things that we were feeding to our herbivorous animals and breaking it into the cell contents and the cell wall. We're tonight focused on the cell wall of the plants that we feed. So that is where the fiber is present. It is a structural carbohydrate. It builds the cell wall. And some of the primary components of that cell wall are the hemicellulose, cellulose, and lignin. And if I just go back a few slides here, I really like this picture here because this shows the hemicellulose and cellulose as fibers, as 2x4s that are really building the structure of those cell walls and give plants the ability to stand up tall and spread their seeds all over the place and be able to send their leaves out and capture the rays of the sun. So that structure is very important, right? And the ways that we analyze this, I think I heard Dr. Williams talk about this earlier, but we have these analyses that you'll hear about, NDF and ADF. You won't hear much about ADL because that goes a little bit nitpicky as far as us nutritionists go. But NDF is neutral detergent fiber. ADF is acid detergent fiber. And all that does is it just refers to the way in which we take a chemical reaction to identify these different components. Okay. And so neutral detergent fiber is a very good measure of all of the fibrous components. It's not perfect, but it is a much better measure than crude fiber. It gives us a very clear picture of all of these fibrous components, of the bulk of the diet. Ruminant nutritionists will oftentimes use NDF to get an idea of how much bulk, how much fiber is there. And as the NDF content gets higher and higher and higher, actually ruminants will tend to decrease their intake a little bit because they get a lot of gut fill. Okay. I'll talk about how things change with plant maturity, but that NDF content tends to increase some as the plants become more mature. ADF, that represents the cellulose and lignin. These are the less digestible components of the fiber. And so as the ADF content gets higher and higher, we see digestibility decline because these components are very hard even for some of the microbes to digest. So if we see particularly high lignin, then we see digestibility that gets much lower. Here's that lignin. Dr. Martinson asked that I talk about this a little bit. Lignin is a fascinating molecule. It is not a carbohydrate, but it's really, I kind of think of it as the glue, a very important structural component that's holding a lot of the fiber together. And as we see more and more of that lignin, the plant becomes more and more woody. It becomes tougher. It becomes less digestible. There's a lot of lignin in the wood that I'm burning in my wood stove tonight. So that is something that is indigestible. We would prefer to see a little bit less lignin in some of the things that we're feeding to horses so that they're able to digest and absorb more of the nutrients that are present. However, that lignin provides important characteristics to the plant. It provides water impermeability and it provides that structural rigidity. So if a plant's going to be able to get water up its stem to the seeds and the leaves, it needs that lignin. If it's going to be able to stand up really tall so that it can get sunlight or so that it can spread its seeds across the field, it needs that lignin. Put together a quick picture so we could sort of get an idea of some different things that we feed to horses here. So this is some data from Equine. It's an interesting, Equine is a forage laboratory that does a lot of analyses, chemical analyses of different feedstuffs that we feed to horses. And what I want to highlight here is NDF. Remember, I told you this is neutral detergent fiber. This represents cellulose, hemicellulose, and lignin. Look how much of the percent of dry matter, that's everything except the water, that all of these different feedstuffs, how much of them is made up by that NDF. So fiber is a very important fraction of our plant material. I mean, we're talking, upwards of 50 to 60% of some of our grasses that are here. And mostly what I'm looking at is grasses. Of course, I think that Dr. Martinson's going to talk some about legumes and alfalfa, and those can be a bit lower. But that's where that fiber is an important component of what we're feeding to horses. I will also note here, I threw oats in just for the fun of it. I want to point out why oats are so special for horses, right? Because they have both the fiber, but notice they also have a lot of this starch. And that starch is a non-structural carbohydrate, but look how those oats stand out from all of our forage sources here. All right. You all should learn, become familiar with plant maturity, understand that as a plant grows, it changes, and the fiber fractions are going to change as that plant grows. Think about how different in regard to what you can see and feel in regard to fiber. Leaves and the stem and the seeds are very different from one another, right? So that stem is really stiff. It's got a lot more fiber in it. Whereas in our young plants, early on, they've got a lot of leaves. They're very soft. They're very digestible. They have less of that fiber present in them. Every one of these plants has their place potentially in some aspect of equine nutrition. And it's important that perhaps you've got young growing horses or mares in early lactation, and you might like to choose a plant that's a little bit more digestible, that has a little bit less cellulose and lignin, and maybe a little bit more hemicellulose and more non-structural carbohydrates, so you can provide more energy. Or like my horse, I've got an old gelding, he's got a bowed tendon, he's not doing anything right now. Well, maybe a more mature forage that has a little bit more fiber would be more beneficial for him. So I want you to have an appreciation for plants being more than just simply a combination of a whole bunch of different carbohydrates, more than just the chemical analysis that we provide to them, but instead realize that they're amazing living organisms that are constantly changing and that are laying down these cell walls that our horses are then making use of as an important energy source, and are also, as we'll talk about, feeding the microbes in their gut. So I want to change gears here and talk about how we might look at the physical characteristics. We've talked about some of the chemical characteristics of that fiber. Let's change gears and talk about some of the physical characteristics. This is one of my favorite slides. And as I look around this slide, these are all the different things that I kind of think about us feeding to horses. What's interesting about this is you can imagine, it's a little bit of a stretch, but you can imagine that in some way, I might be able to design pellets or perhaps a chaff or TMR for horses that had a chemical analysis that was exactly the same as each of these others. So you could have a chemical analysis of all 4 of these that might be very similar if I was formulating those diets. Now, admittedly, that having the fresh forage and the grain, those would be hard to get similar to one another. But I've formulated pellets that are exactly the same as oats. And I've formulated pellets that have exactly the same fiber fractions that we might have in this chaff down here. But we all know that they are physically different from one another. So let's talk a little bit about some of those physical differences. So this is where Jen Weinart and I are working together on some of this. This is work that's been done in dairy nutrition for many, many years, where they understand that dairy cows absolutely need what they talk about as physically effective fiber. And they look at the different diets that they feed to those dairy cows, and they put them on these sieves of different sizes, and they quantify the size of the fiber that they're feeding to their dairy cows. And then they understand that that has a very important impact on the health of those dairy cows and the milk production that they see from those dairy cows. And I'd like to suggest that we can use a similar type of system. Now, there's a lot of work that needs to be done here, but a similar sort of characterization of the physical characteristics of the fiber to understand a little bit more about how that might impact equine health. Remember that first slide that I brought up where we talked about carbohydrates and hydrocarbons? I'm going to keep going there, right? I do like cars and I love horses. And we have many different types of horses and we have many different types of cars. And you better understand about the different fuels that you need. So this diesel truck down here that's in a truck pull, you better not put the same fuel in that diesel truck that you put in your Ferrari F40 because you're going to damage one or the other one. And by the same token, you better not put the same fuel into your thoroughbred racehorse that you're putting into your pulling draft horses. Those horses require different types of fuels, and we can modify the carbohydrates or the hydrocarbons that we provide to them. And in the middle here, of course, I had to put our German. We've got our Mercedes and we've got our nice German warm blood. So we have this whole range, and it's really amazing when you look at the parallels between these and begin to think about how we might change the fiber fractions here and how we might change the hydrocarbon fractions here so that we get the best performance out of these particular vehicles. In order to do that, these two trucks look exactly the same, but I've sort of given away things here. One's a gasoline engine and one is a diesel engine. You can't put the same fuel into both of them, and you have to understand those differences. And so to understand those differences, it's important to look at that fuel intake system. To understand the engine, you have to open up the hood and you have to look and you have to understand a little bit of this. And this is what we as equine nutritionists do with this animal. We've got to understand what's underneath the hood here. How are they utilizing the fiber that we're providing to them? This is from a very famous paper from a woman named Christine Janis, 1976. And this is really a hypothetical picture sort of comparing ruminants and horses, cattle and horses, and how they eat different diets and how they eat the plant material, and how their different fuel systems, having that foregut, that rumen, and digesting fiber that way, whereas the horse, we've got the hindgut and digesting fiber that way, how that might result in some important differences between these two different animals. So I want you to think about the horse's fuel system, and that's what we're going to spend the rest of the talk here sort of talking about, is looking at this fuel system and thinking about how does fiber fit into the way the horse chews things. Look at this, is an incredible fiber intake system right here. Okay, that's what this is built for. I mean, the horse's jaw is an incredible fiber grinding system. The horse's stomach, the horse's gastrointestinal tract, every aspect of this, the form of this really raises a lot of questions about how we can use those structural carbohydrates and the fiber to benefit the well-being and performance of the horse. So let's start at the beginning, right? Horses chew fiber. And there's been some very interesting work that's shown that if we provide that fiber in the form of pellets or hay, and we put markers on the horse's head and we look at the way that they chew, actually the way they chew those two different diets, I guess it's not totally surprising, is completely different. They move their jaw very differently when they're chewing hay versus when they're chewing pellets. It's also important to understand that when a horse chews, it produces saliva. That's important, because that saliva is important in buffering the acid in the horse's stomach. If we even look at the inside of the horse's mouth, and of course, we all understand that their teeth and their molars and premolars are important for grinding, but there are these ridges along the horse's mouth called palatine ridges. And these help to form a bolus as the horse is chewing. This doesn't work quite as well if we don't have that more physical long fiber. I could go on and on about the horse's stomach. The horse's stomach is very unique as far as a mammalian stomach goes, with this very clear dividing line called the margo plicatus right through the middle of the horse's stomach. The upper area is covered by a stratified squamous epithelium. These are layers and layers of cell that are very much like our skin. You know what those are protective against? Those are really protective if you've got a really fibrous diet. The lower region of the stomach is more glandular. This is protective if we have acid. We should expect that the horse's stomach is very well adapted to having more fibrous food up here in the upper regions, and perhaps having less fibrous and more acid production down here in the lower regions of the stomach. And this kind of fits with some work that's been done in ruminants, where on the left-hand side here, we've got the rumen. And ruminant nutritionists are very familiar with the fact that they need a full distribution of different particle sizes in the diets that they're feeding to cows, because they know that they need these fiber mats kind of floating up on top with lower areas where the particles are much smaller to allow the rumen to function correctly. Dr. Al Merritt, who is a leader in the field of gastric ulcers and really did some of the very early and excellent work on gastric ulcers, also appreciated this. And he's developed these ideas and hypotheses about horses eating fibrous diets where they end up having these mats, very similar to ruminants, where they've kind of got some of that longer fiber floating up on top, and even suggests that some of that fiber may be very important for some of the microbes that live there. This is just a picture from a ruminant nutritionist friend of mine demonstrating some of this, or looking at the contents of the cow's rumen. And you can see that that long fiber, it creates this mat that kind of floats up on top. I want you to imagine how that might be very different in a horse that's fed very small fiber lengths. You wouldn't necessarily see that separation of fluid and solid components of things. And this is some work by Marco Lopst and Dr. Nathaniel White at Virginia Tech, who I had an opportunity to work with both of them. Incredible colic scientists and colic surgeons. And they did a little bit of this work where they had cannulated horses. The cannula was actually going into the cecum, and they fed horses different diets, mainly hay-based diet, and then they mixed in hay and grain. And what they found is that when there was a much longer physical length to the the fiber, they got this clear separation of the fluid and solid components. Whereas when they didn't have that, when they were feeding more grain in the diet, and these were really pretty high grain or high concentrate diets, what they ended up with was much more of a frothy mixture that seemed to trap the water and the gases. And they hypothesized that this might be why these diets had a greater risk of colic in these particular animals. So I want you to study and think about the horse's hindgut and look at the complexities of this. This is still an area that's mysterious to us, even as equine nutritionists. I think there's still tons of work to understand exactly what's going on here. As an equine nutritionist, I'm stuck with the job of kind of reverse engineering this and trying to understand, well, what is this system that has adapted over tens of millions of years, and what types of diets is this well adapted to digesting and absorbing and providing energy for the horse? And I would say that this hindgut is very important for fiber. It's just trying to understand exactly how. We all know that an important part of that are the microbes that live in that hindgut. This is from an old slide that I have. Yeah, everybody understands. And you know what? I give a lot of credit to our equine microbiome scientists that are studying this. But a big idea here is that this is an ecosystem. It's a rainforest of all of these different microbial species, that most of those microbes depend on various different forms of fiber as one of their main energy sources. And as they digest that fiber, they're forming these volatile fatty acids that are an important energy source for the horse. So this syntrophy is the idea of all of these different microbial species working with one another to utilize that. And this really highlights one of the other very important places where fiber is important in the diet. It's a feed resource for those microbes. So to summarize all of this, as we think about why fiber is important in the horse's diet, we can move through here, and you should have some new things that you can talk about at the end of this. It's important for stimulating chewing for the horse. It's important as we think about the way that feeds behave in the horse's gastrointestinal tract, forming some of these mats, separating the solid and fluid components of things. It's an absolutely critical feed source for the microbes that are a part of or incorporated into the horse's gastrointestinal tract. If we're not feeding all of those different microbes and keeping a diverse and robust microbial population there, we're certainly increasing the risk of health problems in our horses. That fiber even contributes to some of the mucus layer that helps to protect the enterocytes that line the gastrointestinal tract. And this brings me right back to the beginning. That's what Metamucil does. It's a soluble fiber that actually helps to increase that mucus coating that's very important for the gastrointestinal tract of the horse. So I've got a few more slides here, but I'm running a bit low on time. So I want to highlight the last couple slides. So I'm going to go ahead and flip to those last couple slides. One, I think that the future of understanding fiber will be looking at how much horses chew that fiber, the physical nature of that, and how that contributes to pH through the horse's gastrointestinal tract. And this will really be a challenge for us as equine nutritionists, because we can measure pH in the stomach relatively easily, but understanding how that changes through the rest of the horse's gastrointestinal tract will be a challenge. I'll also highlight here two of my sort of favorite quotes. One from Dr. Harold Hintz, who is, just a god of equine nutrition, and, somebody who I'm glad I had the opportunity to communicate with. And he was there when I was a graduate student. He's since passed away. But it's interesting, in an article from the year 2000, he writes, "Perhaps fiber has been rediscovered. As little as 24% NDF and 14% ADF apparently maintained gut function in horses." So he gave some rough numbers there. It's also interesting to note that in the latest requirements for horses, now sadly that's as long ago as 2007, the line about fiber requirements is this: "Similar concepts have not been developed in horses, but research on the effects of fiber amount and the type on digestion and gastrointestinal health is needed." this is an area of research we really need to do more with going forward. It's still an area where we have a lot to understand. And we're going to hear some a little bit later now from Dr. Martinson about some of that really fascinating research that she's managed to do that I think is really kind of leading our way forward as we understand this more and we look to improving equine well-being and performance. So with that, I will go ahead and stop. I look forward to looking at some of the questions in the question and answer, and also participating in the roundtable at the end of all of this. Again, thank you to Dr. Williams and her team for having me.
Excellent, Dr. Staniar. As always, that was a fantastic talk. Let's give Dr. Staniar a big virtual round of applause. Raise your hand, thumbs up, whatever you want to do, but scream at your computer screen. But that was wonderful. It was exactly what I was hoping to have a talk about today was, you know, what could be more important than fiber in the horse? So I'm going to have Dr. Martinson come on. We're going to get her spotlighted here while she's pulling up her presentation. I'm going to go ahead and introduce her. Dr. Martinson is also a very close friend of mine. She is the equine extension specialist at the University of Minnesota. And I know you have a great following here, Dr. Martinson. You have a lot of Minnesotans following you tonight. So she has been the specialist since 2008. And through that time has led the University of Minnesota's Extension Horse Program and done a lot of applied research that focuses on improving equine forage utilization. In 2019, Dr. Martinson was named the Outstanding Educator from the Equine Science Society, which she is currently the board member. And I did fail to mention that we actually have our president, Dr. Burt Staniar, who was our first speaker. So, so we're big Equine Science Society members as well. I will say, and Dr. Martinson, I have to give it away because I'm sure your Minnesotan followers might be a little jealous that you are presenting outside right now. So that means you are not in Minnesota, but you are where again?
I am in Buckeye, Arizona.
Arizona. So
yes.
I will say I am extraordinarily jealous that Dr. Martinson got to go spend a month horse showing with her two beautiful daughters and three wonderful horses. So, which makes me even more grateful that you took time out of your busy horse show schedule to talk to us today. So I'm going to go ahead and shut up and turn it over to you, Dr. Martinson.
Yeah, thank you so much, Dr. Williams. I'm just going to start my timer to make sure I don't run over on time, but thank you very much for the invitation. I'm happy to be here. I always love working with you in Rutgers. And also, Dr. Staniar, you did a phenomenal job. It's like we practiced and planned, right? But I do also want to say that he is an exceptional president of the Equine Science Society. And if any of you are interested in being a member, we have a virtual meeting coming up. Anyways, just Google it. You'll find we have a great website. So like Dr. Williams said, I'm Krishona Martinson, University of Minnesota, and I am fortunate to be here in Buckeye, Arizona. This is my horse trailer behind me. So I hope the internet holds and I hope there's not too much noise. We'll try our best and cross our fingers. But like she also mentioned, not only do I research horses and forages, but my daughters and I also ride performance horses, mostly timed event horses like barrels and poles and all that fun things. And with everything being remote, we had a wonderful opportunity to spend 5 weeks in Arizona riding with some of the industry's best barrel racers. So it's something that we just couldn't pass up. So Dr. Staniar did a phenomenal job laying the foundation of fiber and why it is so important. And I'm really going to cover 3 projects that we have done that really focus on fiber. It may not be obvious that it's about fiber, but that's really what it's about. So we're going to talk about some of our grazing research. We're going to talk about low lignin or reduced lignin alfalfa. And then we're going to talk about my favorite topic, hay storage and hay waste. So our first project is a grazing project. Like Dr. Staniar said, we all know that horses like grass, and really forages kind of think of 3 different main groups. We have our warm season grasses, and I'm sure those of you in the warmer part of the US or even in that transition zone, maybe type in the, I'm sure you guys, we've been on here for a while, maybe type in the chat, what are some warm season forages in your area? So because of time, I'll just keep on rolling here, but think of some that you guys have access to. In Minnesota, the only warm season grass we can grow is an annual called teff. In the southern part of the US, you guys have Bermuda grass, Bahia grass. So those are the warm season grasses. Then of course we have legumes. Those are things like alfalfa and clovers. And finally, cool season grasses. They tend to grow really everywhere except in the deep south where it's just too hot. So you all can probably rattle off lots of cool season grasses, right? Those are things like orchard grass, brome grass, timothy, ryegrass, lots of cool season grasses. But I think it's important that horse owners know there are some really distinctive differences between these different forage species. And that is important because we want to match our horses' needs with the forage nutritive values of these different groups of plants. So our warm season grasses, they tend to be higher in fiber, right? That's kind of the whole point of today is fiber. They tend to be a little bit lower in crude protein, lower in non-structural carbohydrates, lower in calcium, and lower in digestible energy. And again, these are just some general observations. This is assuming that all of these plants are being compared to one another at the same maturity. On the other flip side, legumes tend to be lower in fiber, higher in crude protein, moderate in non-structural carbohydrates, higher in calcium, and also higher in digestible energy. Again, this is probably a review for many of you. And then our cool season grasses, and I think a reason people like them is they tend to be pretty useful to a lot of different classes of horses. They are moderate in fiber, moderate in crude protein. They do tend to be the highest in non-structural carbohydrates, a little lower in calcium, and more moderate in digestible energy. So we know this, but sometimes we need to kind of validate this, especially in a certain environment like Minnesota, especially throughout the seasons. So the research project that we did had a few main objectives. The first is we just wanted to confirm these nutritive values of these different forages, but we wanted to look at it across the grazing season. So obviously down here, I'm in Arizona, they are cutting alfalfa every day. You guys, it is like a forage specialist dream down here. Every day we cut and bale alfalfa. And it gets too dry, so we actually spray water on it, which is completely foreign to me. But in Minnesota, in New Jersey, and Pennsylvania, we start grazing in the spring and we can graze until the late fall. But do these values change throughout the seasons? And as the horses are grazing these forage species throughout the seasons, does it impact their glucose and insulin response? Really important for all classes of horses, but especially those horses that maybe are prone to metabolic syndrome, have a history of laminitis, maybe they've been diagnosed with PPID, or maybe it's just a super easy keeper where you're trying to keep their body weight in check. So what we did is we had 6 adult horses. Now they were healthy, no signs of metabolic issue, they were at good body weight, and they grazed throughout the entire grazing season, cool season grasses, warm season grasses, and alfalfa. And in Minnesota, our grazing season started in May. That was our spring. Our summer was July. Our fall was September. And then we are actually fortunate that we were able to graze into October, which we call the late fall. And again, I think this is also pretty similar to many people on this Zoom, with the exception if you're in the deep south where you can graze year round. Every time the horses graze, we measured forage quality. We also had catheters in the horses so we could take corresponding measurements on the horses to look at blood glucose and insulin. And also because we had an individual that was really interested in amino acids, we looked at the individual amino acids in the plants and also the plasma amino acids in the horses. So this is what we found. Again, kind of confirming what we had thought. If we look here, hopefully you can see my pointer. CP is crude protein. We know that alfalfa tends to be higher. Now, our cool season grasses in our location are always high. This is oddly high for cool season grasses. We would expect them to be more in that 12 to 14% range, but we always have these really high crude protein values in our forages. We can't really explain it. And then the big thing though, if you look, is we know that teff is going to be a little bit lower in crude protein in comparison. Now let's look at these fiber values. Dr. Staniar did a wonderful job talking about them. NDF is neutral detergent fiber. That tends to be an indicator of how palatable the forage is or how excited the horse is to eat it. And then we have our ADF, or acid detergent fiber, that tends to be an indicator of how available those nutrients are. And what you can see, and if you look at these little letters behind each of the values, if the letters are different, it means that the values are statistically different. If they're the same, it means that even though they are numerically different, they are statistically the same. So the big take-home message here is that we can see that teff has higher values of NDF and ADF. So they're going to eat it a little slower and the nutrients aren't going to be quite as available as compared to something like alfalfa. Now that doesn't mean it's bad, it just means use that to your advantage. If you have a horse that eats super fast, or a horse on maintenance, or an overweight horse, use that to your advantage and maybe select something that's a little bit higher in fiber to slow them down and make the nutrients not as available. The other thing we really like to look at is non-structural carbohydrates. So right, ADF, NDF, NSC, those are all fibers. It's just that NDF and ADF are those structural, where the non-structural carbohydrates are the ones that I think get a lot of attention, but they're all equally important. And as we knew, our cool season grasses tend to be higher, legumes tend to be intermediate, and our tough grass or our warm season grasses tend to be lower. And then of course, when we look at everything holistically, we know that alfalfa is more energy dense. That's why we feed alfalfa to performance horses. And we know that teff and the cool season grasses are going to be less energy dense and are probably more suited to horses in light work, maybe moderate work, or those easy keeping horses. So how did these change throughout the seasons? So here is our neutral detergent fiber. And we have on this axis, we have spring, summer, fall, and late fall. So in Minnesota, this was May to October. Now, we can't even plant teff until May because our soils are too cold. So we didn't have teff to graze in the early spring. We only had the alfalfa, which is these green dots, and the cool season grasses, which are blue. Now in the late fall, the alfalfa had already gone dormant, but the grasses were still growing. But if we look, you can see that there were differences between the grasses, but in general, during the spring or during the summer and fall, teff tended to have higher values of neutral detergent fiber, where in the late fall, it was very similar to cool season grasses. And of course, when the alfalfa was actively growing, it was always less. So you do see a little bit of a difference throughout the seasons. Again, it's just something to keep in mind, something that I thought was of interest. Now, the one that really was of interest, and our colleague, Dr. McIntosh, has done great work in this, was the seasonal variations in non-structural carbohydrates. So again, we can see that the tough grass, those warm season grasses, was lower, especially during the fall and the late fall, and especially compared to our cool season grasses. So we know when we have cool nights, warm summer days, especially when those nights in the fall and late fall get above freezing, we know that cool season grasses can accumulate a lot of non-structural carbohydrates. It's just the mechanism and how the plant accumulates it. Legumes and warm season grasses really can't accumulate as much as a cool season grass does. So you can see we had fairly big differences in the late fall because we had the right environment and we had these differences in these species. Now, all of our horses were healthy, so none of this created any problem. However, if you would have a horse that is prone to laminitis or has a history of laminitis, is diagnosed with metabolic syndrome, has PPID, or maybe is an easy keeper that you're really watching, the fall and late fall is when you really, really want to pay attention to what you are grazing. And of course, year long, what you are feeding in the form of hay. And that is why we do watch those diseased horses more carefully when we get those big swings in non-structural carbohydrates. So how did this translate into the horse? So remember, every time we graze the horses, we would take samples of the forage. We would also take blood from the horses to see how it was impacting their insulin and glucose. And let's just look at insulin. So we did look at the horses fasting. So that's those little gray circles that are new. But when we look, when the horses were grazing, we can see that their average insulin was higher when they were grazing cool season grasses compared to teff, especially in the fall and late fall. And again, that is just reflective of those non-structural carbohydrates and probably some of the other forage components that maybe aren't as obvious as non-structural carbohydrates. So again, that increase in non-structural carbohydrates is reflective and mirrors that increase, or maybe is a result of, that increase in the insulin. So again, just something, to keep in mind. These were healthy horses. We saw no issues whatsoever. This is something to be aware of if you have horses that have one of these diseases where insulin needs to be watched really carefully. So the other fun thing we did is, because we had a muscle biologist on the team, is, these warm season grasses are great. I know those of you in the southern US, Bermuda grass, Bahia grass, you have access to it. In the north, we don't have warm season grasses really until we started working with teff, which is an annual warm season grass. So when we are trying to reduce the non-structural carbohydrates in cool season grasses, one of the only things we can do is soak hay. And when it is 30 below out, soaking hay in your bathtub and schlepping it out to your barn is horrible, right? It freezes, it causes a hot mess. So we're trying to figure out, okay, can teff be something we use to just lower the non-structural carbohydrates, which is great, and it does, and we know it does, but the crude protein can get pretty low. And I know we can supplement crude protein, but we wanted to make sure that we weren't going to cause problems by recommending a forage by focusing on just one thing, like non-structural carbohydrates. So we thought, okay, it's lower in crude protein, but these are adult horses at maintenance. What does it really look like? So here is the amino acid profile of these different forages in our grazing trial. And crude protein really is just kind of an indicator of amino acid. Amino acid is what you really want to look for. Now, we don't balance horses' diets for all of the amino acids, but we will probably get there eventually. So you can see there's differences, but when we statistically looked at it, the only statistical difference we had was in leucine. Okay, so how does this translate into the plasma amino acids of the horse? We thought, oh my goodness, like teff looks kind of lower. Are we going to have all of these problems in the plasma of the horses? Well, we didn't. So yay, right? We did have one difference between teff and cool season grasses, but only with valine. So, is teff going to be an issue with its lower crude protein? Probably not. But with any forage, you should have it tested, and you should watch, especially if it's the sole or primary source in your horse's diet, because of how it is harvested. You saw that in the Stanley commercial. Dr. Staniar talked about the maturity, just the soil fertility, the physical environment. But again, if you are doing a good job of harvesting and growing that forage, the amino acid in teff shouldn't be an issue for that adult horse at maintenance. Now, horses in heavy work, that is a question for Dr. Williams and Dr. Staniar. I'll put that on them. So really the take-home message with this project is we were able to confirm that teff as a cool season grass was lower in non-structural carbohydrates and higher in those fiber concentrations of ADF and NDF in comparison to cool season grasses and legumes, but we kind of already knew that. Horses grazing teff did have a consistent consistently lower insulinemic response, especially in the fall and late fall. And we were excited to see that because sometimes it's really nice to give these metabolic horses some grazing time and something that will have less of a negative impact on them. But the really fun thing we found out is that even though these cool season grasses and alfalfa were significantly higher in crude protein, the individual amino acids were rarely different, and rarely did that have an impact on the horse's plasma amino acid concentration. So again, those adult horses at maintenance were getting enough. How that translates to the equine athlete, I guess that's a study for another day. So transitioning to the next project that was fun, being a forage specialist, we've been working a lot with reduced lignin alfalfa for exactly the same reason that Dr. Staniar pointed out. Lignin isn't digestible to the animal, but of course it's very important to hold that plant upright. But plant breeders have been doing a good job, especially when it comes to dairy cattle and ruminants, cattle and sheep, is they know small reductions in lignin in alfalfa really results, they can see it in increased milk production and increased meat production. So it is something that we wanted to see with horses, and we have been doing a lot of work with reduced lignin alfalfa in the field, so we had access to this hay. So what we had found previously is that we knew that the reduced lignin alfalfa was actually reduced lignin, like they said. And in the cattle studies, it did lead to increases in vitro dry matter digestibility and neutral detergent fiber digestibility. So that's really promising. So what we did is we actually used a protocol that Dr. Staniar developed, and we just followed it exactly to the T. So thank you for that. We evaluated the apparent digestibility, and you guys, when Dr. Staniar was talking about the fiber particle size, we looked at the back end. We looked at, was the feces, using that Penn State shaker system, was the fecal particle size different when feeding reduced lignin and non-reduced lignin alfalfa? We looked at that mean retention time. We can look at that by feeding horses non-digestible markers. And we did this again with these same adult horses. So again, we fed reduced lignin and non-reduced lignin alfalfa hay to 6 healthy adult horses. This is Dr. Staniar's protocol. They had a 9-day adaptation phase, followed by 5 days of total fecal collection. And yes, it sounds just like it is. You collect everything the horse puts out for 5 total days. They were fed markers to determine the liquid in particulate or solid mean retention time. So how long does that forage, does that feed take to go from when the horse is eating it to when it's excreted? We then looked at dry matter digestibility, retention time, fecal particle size, and because we had a new faculty member that was interested in microbiome, that was his background, he actually could extract DNA from the feces, and he could look at the microbiome differences. So like Dr. Staniar said, microbiome is just like, this is like the initial frontier of that research. It's super exciting to be on the front doorstep of that research. So this was the hay that we fed. And again, we had been working with this hay and doing other projects with it. So we knew we had a reduced lignin alfalfa and a non-reduced lignin alfalfa that were very, very similar, except for the lignin. So the crude protein of both of them was about 20%. ADF and NDF, again, those fiber values were very similar. ADL is acid detergent lignin, or the measurement of lignin. And again, these are significant differences. You may look at that and say 8.1 and 7.4, like that is nothing. You guys, when that comes to lignin, that is huge. So that is a significant difference. And again, the digestible energy was also the same between those forages. So the only thing that was different was the acid detergent lignin. So how did the horses eat it? So of course we fed the horses, we fed them out of hay nets. Their dry matter intake was for both, like they ate them very similarly. And this was important to document all of this because again, we're looking at digestibility and how fast these different feedstuffs move throughout the horse. They both ate about 1.6% of their body weight. Dry matter intake rate was about 0.7 kilos per hour. And we also measured water intake because we were looking at how fast that liquid moved throughout the digestive tract. And they ate just over 6%, or they drank just over 6% of their body weight. So all of this, I don't know how Dr. Staniar did this, because this is incredibly labor expensive and very expensive research to do. But interestingly enough, when we fed the horses reduced lignin alfalfa, their dry matter digestibility, or DMD, did increase 2 percentage points. That was significant. We were excited. The mean fecal particle size, the particle sizes of the feces. So yes, we collected the poop, we dehydrated it, we mixed it up, and we put it in those Penn State shakers, and we shook the feces, and we measured all of the different lengths that it came out of. And it is about as exciting as it sounds. There was no difference. There were numerical differences in the liquid mean retention time and the particulate or solid mean retention time, but they were not statistically different. So anywhere from 22 to 26 hours for that liquid to go through the animal, and 25 to 31 hours for that particulate to go through the animal. And if you want to look at this, I just think these figures are amazing, right? I think it's so interesting to see how it moves through the horse. So if we look back 1 second, we can see that maybe, that reduced lignin alfalfa appears to be moving more quickly through, but it isn't statistically different. So the reduced lignin alfalfa is the blue line, where the non-reduced lignin alfalfa is the red dashed line. And remember, Dr. Staniar said it takes about 48 hours for that feed to move through the horse. Well, look at this bell-shaped curve. He's right on the money, right? The liquid about 48 hours. And again, that particulate, you can see the peaks are a little bit different, but again, they're not significantly different between the alfalfa types. But I just think it's fun to see how things move through a horse. Like Dr. Staniar said, you almost have to reverse engineer this stuff because the horse is the horse, and we have to try to figure things out. So then we had our microbiome faculty member come in, and it was just so fun to work with him and to see what they did. So again, we can take the feces and we can extract DNA from the feces quite easily. And we can use that to look at the diversity of the microbiome in the feces. And what we found is that it didn't matter when we looked at the herd. So the circles on this figure represent horses' microbiome diversity being fed the control or the non-reduced alfalfa, and the squares is the reduced lignin alfalfa. And again, there was no impact on a herd basis. So all of the horses, you can see, it's just scattered. But when we boiled it down to the individual, and any of you that have more than one horse know that if you have a herd of 4 horses, my goodness, they are individuals. If you could just manage them all the same, wouldn't that make your life easy? But no, that's just not how it works. When we looked at the horses individually, their individual microbiomes changed with the diets, and we could watch it change. So if we look at this figure down here on the bottom, here's the time point 1. So the point when we started collecting the information from the horse, we can see here that when the horse is being fed reduced lignin alfalfa, its microbiome diversity was kind of in this quadrant. And then when it switched to that control alfalfa, or the non-reduced lignin alfalfa, we could see its microbiome shift. And the only difference was that 0.5% difference in the acid detergent lignin, right? Because we knew everything else was the same. We knew the feces, we knew the feces particles were the same size. We knew that it was traveling through the horse at the same rate. So you guys, this is so cool. But also when I see this, I think, man, if you are switching diets, you better do it really gradually. Like when your nutritionist or your veterinarian says, change diet slowly over 14 days, take that advice incredibly seriously and do it slowly over time, because we don't want these little microbes that are tasked with breaking down and digesting these fibers to die off quickly because of this rapid change, because that's when we run into problems with colic and founder and the things that that send fear into the minds of any horse owner. So this was like a 2-year-long project, and I'm going to boil it down into 4 bullet points. And it makes me kind of sad. It's only full 4 bullet points, but I guess it's good, right? So the reduced lignin alfalfa hay does have a lower amount of lignin and a greater apparent dry matter digestibility. So it is more digestible to the horse. The horse's gut microbiome responds in an individual manner to very small changes in lignin. And again, I cannot emphasize it enough. You need to make these changes really, really slowly. And again, you know, I think about this as a bigger picture. Horses tend to be individuals, right? We manage them as individuals if we can. I would love to manage all of my horses as a group. It's just not going to happen. Now, there is a practical question that I don't know how to answer. And does reduced lignin alfalfa really have a place in the horse industry? You know, maybe for that older horse who is struggling to keep weight on, or that performance horse that needs more energy. Does that greater dry matter digestibility really impact the horse? And we haven't taken that next step yet in the research process, but sometimes we have to take baby steps and answer questions along the way. We can't answer the big question right away. We have to work our way up to it. All right, you all probably like, whew, that was a lot. That was a lot. So, but it's exciting, isn't it? Like all these new things that we find and that we can discover. So I'm going to just spend the last few minutes, and Dr. Williams, I will not go over time. I will make sure of that, talking about one of my favorite topics, and that is storing round bales and really hay waste. So I think horse owners have a love-hate relationship with round bales, right? You love them because they're convenient. They tend to be less expensive, but man, there can be a lot of waste with storage and feeding. So we know from past research that when you store round bales outside, we can have 7 to 35% waste just from weathering. Now, how those round bales are bound makes a difference. So in the upper left hand here, we have what is known as net wrap. It almost looks like a fishing net kind of put around the hay. Usually there's a couple layers on it. So the hay can breathe, but it has some coverage. In the upper right-hand side, we have twine. Again, it tends to be kind of reddish or greenish. And technically that twine is supposed to be spaced out every 2 inches. There's a new product called B-Wrap, which is here in the lower left-hand side. And we've been doing some research with that. And that is like a Tyvek that is held in place with net wrap. So it really gives that bale complete coverage, but it's not that plastic wrap or those kind of white marshmallows that people call them that you see in the fields. And we know that, Dr. Williams, am I still live? I just got a reading saying that my internet was unstable.
Nope, you still look good on my end.
Okay, thank you. So we know that twine bales, because it doesn't have as much coverage, we can have more waste than with net wrap, but we're really not sure about B-Wrap. And of course, we've all seen this image in the lower right-hand side where we have that weathered layer, or how those round bales look when they've been stored outside. So what we did is we baled hay, we baled alfalfa with twine, net wrap, and B-Wrap, and we stored it outdoors for a year. And every 3 months, we took samples and we weighed the hay to see if there was going to be a big difference. And there was a big difference. So the big thing that we did differently, and it was out of necessity, if you are in Minnesota or Pennsylvania or New Jersey, if you get any kind of freezing rain or heavy snowfall and you do not store round bales on pallets, when you go to pick them up, that binding material freezes to the ground and you lose half of your bale. So we stored our bales on pallets because we knew every 3 months we had to lift them up, sample them, and weigh them. Because of that, we actually had less dry matter loss than previous studies. So our twine bales only lost 7% dry matter, net wrap 5%, the bee wrap lost none. And dry matter is important because that's what you're feeding. You want to retain that dry matter. So the other interesting thing that we looked at is in this figure over here on the right-hand side. You can see this little dash line on the bottom. This is the bale moisture of bee wrap throughout that 1 year. And the harvest happened in June. So again, every 3 months until the following June. The gray line is the net wrap, and this the lightest gray line at the top with the triangles is the twine. So you can see that we had really great luck. The round bales were baled at about 10% moisture, but after 180 days in storage, those net wrap and twine bales were at 24% moisture. We know from previous research that round bales need to be kept at less than 15% to not have mold formation. So anything over 15%, and we are going to get mold formation in a big bale package like that. With small square bales, we can go up to 17% moisture usually. Again, just because we don't have 900 pounds of hay, we have 60 or 80 pounds of hay. So again, it's a package size moisture. But you guys look at the moisture. Oh my goodness. That's how much moisture those bales picked up from the environment. But interestingly enough, that kind of brown, icky weathering layer that we saw still did not go beyond 6 inches, even with the twine. And we know that because we actually stratified our forage samples where we had forage quality of the first 6 inches and then from 6 inches into 18 inches into the bale. So how does that translate? How does that tie into fiber? So interestingly enough, I mentioned in passing that non-structural carbohydrates are water soluble because we can soak hay to remove them, but don't ever soak hay for more than an hour. But we can soak hay to remove the non-structural carbohydrates. So when this hay is getting rained on outside uncovered, when you have snow, we have the freeze thaw that we have, those non-structural carbohydrates are actually being dissolved. Now on the flip side, the components of fiber that make up neutral detergent fiber and acid detergent fiber are not water soluble. So because they cannot be dissolved by the environment, by the moisture, they become concentrated. But we know that they are not digestible by the animal. So hay that is improperly stored and has a lot of waste, what we see is a concentration of these fiber components, ADF and NDF, that aren't digestible to the horse, and we see a leaching of our non-structural carbohydrates. So after 12 months in storage, we actually waited another 4 months, and then we fed this hay to cattle. We couldn't feed it to horses because you'll see the mold count issue, but we could feed it to cattle. At that time, everything had kind of settled and neutralized, and there wasn't a huge difference in crude protein or moisture, but we did see this concentration on the NDF and the ADF. Again, it was small concentrations, but we saw it. So again, improperly stored hay that's been exposed to moisture will have a concentrating effect of these fibrous components that aren't digestible and a leaching of our non-structural carbohydrates. But the big kicker, you guys, was in mold. So our current recommendation for mold is we want all horse feedstuffs to be less than 500,000 colony forming units. So sitting outside here in beautiful sunny Arizona, whether in your home office, whether wherever you're at, your living room, there's some mold in the air. You can never be at zero. We want anything fed to horses to be less than 500,000. We can feed cattle up to 1 million, maybe 5 million if we're mixing it. Those twine bales, remember how moist they got? Remember the moisture around 180 days? 7 million colony forming units of mold. Again, it was concentrated that outer 6-inch weathered layer, but it was very moldy in that outer 6-inch layer. The net wrap was at about 4.7 million, and that bee wrap, because it didn't have the moisture penetration, was only at 48,000, so well below our 500,000. So B-wrap appears to be a good option. It does pay for itself, but you do have to store the hay outside more than 90 days. So again, there are some caveats, but it's just an interesting fact. So we've talked a little bit about storing. How about feeding? So one of the favorite research projects I ever have done, but it was horribly labor intensive, is we actually looked at 9 different round bale feeders and a no feeder control. So we put out these round bale feeder, we put out round bales in feeders, and then just put out a bale shown here in the lower left-hand corner and let the horses eat. We weighed the horses going in and out of each pen. We weighed the hay, we knew the bale weight, and every single day we would go out there and rake up all of the waste that was on the ground around the feeder. We would dry it because of course it was full of feces and urine and mud, dry it, clean it, and then weigh it. And then we can estimate how much waste. So these are the round bale feeders. If you Google University of Minnesota and round bale research, we have a web article that will come up as well. So the take home message in this whole thing is, please, please, please use a feeder. I don't care what feeder you use, just use a feeder. So our no feeder control, when we just plop the round bale out there, 57% waste. You guys, over half of your hay is being pounded into the mud and making manure that you then have to pay to have removed. Some of our more efficient round bale feeders were around 5 to 6 to 9% waste, so a dramatic reduction. Our circular feeders were about that 19% waste. So no feeder is going to give you 0% waste. It just doesn't exist. But we can dramatically cut down the waste. Over here is the cost of the feeders at the time of the research. These costs may fluctuate a bit, but hay price at $200 a ton, which is pretty average right now for Minnesota. I realize in other parts of the country, it is dramatically higher, but again, it's all in perspective. All of these feeders paid for themselves within 10 months. So in my opinion, you cannot afford to not use a feeder when you are feeding round bales. Now, this study was very eye-opening to us. It was very popular. So we followed it up by looking at feeding small square bales. We used a basket feeder, a grazer, a porta grazer, and then this rack feeder. And of course, we also fed hay on the ground. And we saw the same thing. When we fed small square bales on the ground at 2% of the herd body weight, we had 13% waste, and the feeders were anywhere from 1 to 5% waste. And again, all of the feeders paid for themselves within 1 year. If we look at this in a larger setting, there was a great research done in Texas where they fed small square bales in a stall. Very similar waste to when we fed small square bales outside, about 1 to 5% with feeders and about 13% without a feeder. So we use these values when we looked at if you were feeding 5 horses without a feeder in a box stall, outside, or feeding round bales. If you're feeding those 5 horses, and this is assuming they're 5 1,000-pound horses that are being fed a primarily hay diet, and hay valued at about $250 a ton. You know, we do have some hay waste. You just can't get away from it. But look at the difference. If you're feeding small square bales indoor or outside, $56 in waste versus $731 without a feeder. Now, if you're at a mid-sized facility with 15 horses, you know, let's look at the round bales. We're still going to have about $844 of waste, but without a feeder, that goes up to $9,600. If you're at a very large facility, let's say you're feeding without a feeder in a box stall, you are still wasting $4,000 a year. That buys a lot of feeders. And again, it doesn't even matter what feeder you use, whatever one you prefer. But if you are feeding a large number of horses without a feeder, especially round bales, $32,000. That is, you could buy round bale feeders till you're blue in the face. So the take-home message from this is that moisture penetration, when we are looking at the different types of hay, you know, the binding, whether it's twine, nut wrap, or bee wrap, the most important thing you need to do is to keep the moisture out. because again, you want to limit the concentration of those fiber components and the leaching of those non-structural components. And then again, just please, please, please use a feeder when you're feeding hay. Any feeder, I don't care, just please use a feeder, especially with the round bale. So with that, this research was really the results of 3 tremendous graduate students, Amanda Grev, Michelle DeBoer, and then Amanda Ryder. And with that, Dr. Williams, I will turn it over to you.
Oh, excellent, Dr. Martinson. Thank you so much for that. Let's give her a nice big round of applause, virtual round of applause. Raise your hand, thumbs up again, however you want to do it. So what I'm going to do now, there's 2 things I'm going to do. First, I'm going to ask Kyle or Jen to go ahead and pin all of our speakers so we can get our Q&A panel ready to go. And while they're going ahead and pinning all of us to the screen, you guys can turn on your videos for me. I want to do one thing because I know it's getting a little late. I still want to answer a bunch of your questions that have come up, but if you do have to log off, I totally understand, but I want to ask you to do one thing for me really quick. It'll take you about 3 minutes. I'd really love you to evaluate our program and evaluate the speakers and the topics and everything. So I'm typing into the chat now a link. If you would go to that Go Rutgers link, it'll take you to a survey. If you've been on the last 2 webinars and you fill out the survey, you could do it again. It'll ask you the same questions, but it's about these particular speakers and these topics. And I really want to know if there's anything that you want to see in the future, what you liked, what you didn't like, etc. So please go ahead and do that. So we're going to go ahead and pull open the Q&A box. And I know both Ellen and Jen have been busy, and I know Dr. Staniar has really been busy answering a lot of questions, but I think we still have more come up. So I'm going to let Jen take over and start asking us questions. How's that sound?
That sounds good. We have a lot of questions. Some of them are about your research that you presented, and some of them are more general questions about forages, and others are more general nutrition questions. So we're going to try to mix it up a little bit here with the time that we have left. I'm going to lead with this question. Why are so many horses becoming metabolic or insulin resistant and sensitive to grasses compared to 40 years ago? Horses used to only founder if they got into a bin of rich grain or a pony turned out on rich pasture. What has changed in their bodies that is increasing inflammation and increasing their sensitivity to sugars?
I'll take a first shot at that. Dr. Martinson's been talking for a while. You know, I'll tell a really interesting story. We have another very accomplished researcher here in Pennsylvania, Dr. Sue McDonald, who does a lot of work with behavior and with ponies. She's at the University of Pennsylvania. And she and I had a conversation once, and she was telling me about her pony herd, her semi-feral pony herd, that she sees very little laminitis in that group of ponies. However, when she sells those ponies, because she has to sell some every year, and people here in Pennsylvania in the Northeast region buy those ponies and they take them home, all of a sudden those ponies, some of them start to get laminitis. We all know ponies are sort of at a high risk for insulin resistance and metabolic syndrome and laminitis. And one of the things she noted is that those ponies in her semi-feral herd, every year they kind of, particularly when the grasses get rich and that sort of thing, they're all running around, and it happens to be the reproductive time of the year when the stallions and the mares are all running around after each other. And there's a lot of exercise that's going on. I think that we're not quite, we're probably doing all right on the nutritional side of things, but we may not be doing well on the exercise side of things. I think that those fat little ponies do well when they're exercising a lot and they're burning off a lot of that energy. But when they're standing around super happy in their field in April and they're just eating lots of non-structural carbohydrates, that becomes a metabolic issue for them. So I think that it takes a broader perspective of that as to, you know what, 40 or 50 years ago, we were probably working many of our horses more than we are today. And that may have some impact on the amount of metabolic problems that we see.
Great, thank you, Dr. Staniar. So following up on that just a little bit, there was a related question, and I don't know if there's anything out there that has specifically looked at this, but I think possibly. But do you know anything about the relationship between horses' systemic inflammation levels and different forages?
I will say that what is fascinating about the inflammation and the immune system is that immediately below the first layer of cells that are in the gastrointestinal tract, the immune system is right there. And there are, in fact, very specialized cells, immune cells called dendritic cells, that, believe it or not, actually reach a little hand, a little part of themselves. Can you hear me all right, Jen? You look surprised. Everybody else can hear me. They're nodding their heads. These dendritic cells actually reach up through the gastrointestinal tract and reach out into the lumen. So it is absolutely possible that the fiber that we're feeding and the feeds that we're feeding have a direct impact on the horse's immune system. And those dendritic cells actually impact the inflammatory response as well. So we're understanding that more and more, and there's good evidence that yes, that probably has a direct impact on the immune system. There was actually another excellent question in the question and answer about allergies and how leaky gut might influence allergies. So I think that was right in line with that as well.
Yeah, and actually, Dr. Staniar, I'm glad you brought up allergies, because there's kind of been some questions that have all kind of targeted one thing. And I know me and you went back and forth on a question and a case a couple months ago, and I reached out to all of my fellow nutritionists because I seem to get it a lot this time of year about horses, particularly older ones, but I have heard it in younger ones, that get that diarrhea squirts, kind of leaky, squirty. They might have normal feces, but it's very liquidy either before and after. And I've heard people say, is it allergies? Is it the diet? What can we recommend? And I know you gave a really good recommendation when I reached out to you the last time. Do you mind sharing?
I don't remember my really good recommendation, Dr. Williams. That's not right.
Well, let me give you a little more on that one, because I thought it was really interesting too. Because working with this particular individual, it sounded like she had done everything I would have recommended, which is basically change the forage and, look at different hay sources and all of that. And really, you just talked about a lot of what your talk did is about making sure the horse has a bunch of different fiber lengths and sources, and not just one type, but make a variety of carbohydrate sources. And I know you say it better than I do.
I love that. And so this actually gets at something that Dr. Martinson brought up, because one of the points that Dr. Martinson brought up, she said, well, I don't really know about the place of this lower lignin alfalfa and how important it really is for horses. Gosh, I would love to have one more forage source that was a little bit different than all the others. I think that the as a nutritionist, I think of everything in moderation. I think of the variety in the diet. And I think that that's very true of all the carbohydrate fractions that we're feeding. So I do very much think that the more that we can feed our horses a wide range of different fiber sources, the better we're sort of supporting a very diverse microbial ecosystem, a robust microbial ecosystem that is able to adapt to different situations. So for example, here in Pennsylvania, we have many people, it is sleeting and icing, and there's 12 inches of snow out there. Most of the smart horse people have decided to go down to Aiken, South Carolina or Florida at this point, but they're going to have different forage sources down there. If they've fed their horse kind of a very flexible, wide range of different carbohydrate fractions, their horses are going to be able to better adapt to the change in the diet that they're going to have down in Florida or down in Aiken, South Carolina, of forage sources that they're not going to get here. So that can help somewhat. I did reply to another person in the question and answer. And, the fact of the matter is, diarrhea is tough. I don't have all the answers for that. I have some ideas and some things that I can suggest, and I like talking to individuals and trying to help them with those problems. But they're just, sometimes I will say that sometimes I can't solve it. I don't have the solutions. Those are really hard. And sometimes actually the problem is with the horse. I've tried to solve problems. Sometimes there was a horse that passed away about 2 years ago, and I was trying and trying and trying from a nutritional standpoint. And it turned out that they actually had cancer of their gastrointestinal tract, and that's why they had diarrhea, and they finally ended up dying. And we looked at the gastrointestinal tract. There was nothing I could have done from a nutritional perspective, but I tried. But sometimes you just can't solve that problem. It's just really hard.
Yeah, and I know it's frustrating. I saw a question come up asking if we own horses, and we all do. So it's frustrating too when your own horse has that problem that you can't solve, because literally my horse was one of those that had the diarrhea, squirty, whatever's, and we tried everything under the sun to get it to go away. And all of a sudden, spontaneously, it just went away.
All right. Well, I'm going to throw some questions back to Dr. Martinson. We've had a lot of questions come into the Q&A about the slow feeders and feeder recommendations and feeder design. And a couple of the things that came up were specifically, if you have a horse with heaves, what do you think the best feeder is? And then also, how do you see slow feeders as an aid in digestion? Like, how do you see the relationship between digestion and slow feeding?
Sure, great. So first of all, with the horse with heaves, I think it's incredibly important to work with your veterinarian to make sure that you are following that advice. With heaves, the most important thing is to control the dust. So that's not really a soaking issue. That's more of just wetting it down. And then again, any type of feeder that would not allow that horse to bury its nose into the hay. So the slow feed hay nets, any of the hay feeders that have any kind of barrier between the horse and the hay, where they can still eat it, but they can't bury their nose in it. So again, it's about dust control, usually with the horse with heaves. So we've done some projects with slow feed hay nets. I'm a huge fan based on what we have found. So we know that when we feed horses 1% of their body weight, so with an average 1,000-pound horse, that's 10 pounds. When we feed them 10 pounds of hay on the ground, they can eat it in about 3.5 hours. When we feed it from a slow feed hay net, which has 1 1/4 inch triangular openings, that goes to 6.5 hours for that exact same hay meal. And like Dr. Staniar shared, the horse only produces their saliva, that buffered saliva, when they chew. So a horse in a natural setting will graze for about 14 to 16 hours a day. So if we can simulate that grazing, and we can have them eat little small meals throughout the day, but not feed them brown bales so they get obese, but feed them a controlled diet in a slow feed hay net, I think that's beneficial. Now, are hay nets good for every horse? No. If you have an older horse, a horse that you're trying to get a lot of feed in or forage into at quickly, or a horse that's a picky eater, not great. But for a lot of horses, I think it is a great tool to have in your toolbox.
Thank you, Dr. Martinson. And also, I see that we're we still have a couple of questions we want to try to get in there, because we do have some people hanging around, but we also have some people that are logging off. So before we lose before we lose all of our attendees, I would like to just thank you all for making this such a great seminar series in general, and a great event tonight. And Dr. Williams just put the link for the survey again in the chat. So if you have a chance, if you could please take a moment to fill that out, we would really appreciate it. I think we're going to take 2 topic areas to close out here. And the first one that we've had come in a lot are questions about winter feeding. And so the first one I'm gonna start with is someone who said their barn has had a lot of colics recently, but all the horses were on different diets, different hays. And do you have any thoughts on adjusting winter feeding to reduce incidence of colic?
We're all looking at each other. Colic, I will say, because, and then I'll let the speakers take it, but colic to me is very similar to diarrhea. It's not everyone has the answers, and it is a really tricky thing. So it's definitely not my topic area. Like Dr. Staniar, you said, Dr. Lopes and Dr. White were doing so much with colic. I don't know if there's anything from their research that kind of were a take-home message for you.
Colic needs to be more clearly defined, particularly in a barn where there's a lot of colic going on. So for example, in my nutrition class today, we were actually talking about colic, and I asked the students, what they think about when they think about colic. And one student said impaction colic, and another student said gas colic, and another student said torsional colic. Another student asked about ulcers. Colic in general just defines abdominal pain. And so there are many, many different types of colic. The horse's gastrointestinal tract is very complex. And so, if all the horses, for example, if all the horses are dealing, if it's always impaction colic, if every single horse is getting impaction at the pelvic flexure, and that seems to be the problem, then you might look at the digestibility of the forage that you're providing, because perhaps there are some issues with it being a very mature, high ADF forage that the horses just are having a difficult time digesting, and maybe they're not getting enough fluid intake. It's winter, they're not drinking as much, and so that's causing some problems. But it could go totally the other direction, and you might have something in the diet that was resulting in a lower pH or more acidic environment and more gas production. And therefore, then you're dealing with more of a gas colic type of problem. And so it's impossible to answer the question as asked. But I think exploring a little more closely, okay, what are the similarities? And really, Dr. Martinson said it, working very closely in kind of working up that case with your veterinarian, exploring all of that, and playing a little bit of a detective in trying to figure out what's going on there. As asked, there's not a real clear answer to the question.
Great. So more in general then, how do you determine what's best to feed horses in cold temperatures versus warm temperatures?
Dr. Martinson, when you are at home versus in Arizona, do you feed your competitive trail horses any differently?
Yeah. So that's a great question. And I think one thing that we struggle with is people think they need to feed more grain, but I'm telling you, please, please, please. So we know that when temperatures dip below 18 degrees Fahrenheit, which I think is all of the country except for where I am at in Buckeye, Arizona, when the temperatures dip below 18 degrees, we know that horses need a little bit more energy to stay warm, right? They need that. And again, I'm very aware that I'm speaking with two nutritionists who are trained in this, Dr. Williams, Dr. Staniar, on the Zoom, but the heat is produced when the forage is fermented and digested, right? That is what produces the long-term heat. So we want to give the horses a few more pounds of hay anytime that it is below 18 degrees, and that is per day. Of course, between 0 degrees, this is Fahrenheit, and 18, maybe a pound. I think, Dr. Williams, you might know the exact calculation off the top of your head. But obviously, I mean, it has been negative 35 back in Minnesota. It has been super cold for like 10 days. At that point, if you have horses that are outdoors, even if they have access to shelter and as good quality forage, they have access to water, maybe they're blanketed, in that extreme of weather, they almost need kind of continuous access to forage to help keep them warm. But with that, I will turn it over to the true nutritionist who can maybe add anything there.
Hey, I can't agree more. I mean, hay is where it's at. You really need to provide them a lot of hay. And I actually just saw a comment come through too that I can imagine with temperatures that cold, which we haven't had it that cold in New Jersey yet this year. I can imagine drinking water is even an issue too, right? I mean, just that cold, not just the frozen water, but the temperature of the water.
Yeah, we don't want to see water get much cooler than 45 degrees Fahrenheit, which in those temperatures, you have to pull out all the stops, right? We're talking like insulating, putting hair dryers, putting in light bulbs, getting it out of the wind, anything you can, obviously tank heaters, whatever you can do, but it is extremely challenging in those environments. But the most important thing is just making sure that the water does not freeze, right? Like they can't rely on snow. But yes, Dr. Williams, you're absolutely right, is water consumption during that really cold temperatures.
Well, I actually think that that's a good place to end. I know we have a few that we didn't get to, and we'll try to put them in our Equine Science Center Ask the Expert column, and we'll try to get to them. But on that note, I need to go get a hot cup of hot cocoa or something, because it's actually making me feel a little chilly. I will say, as Jen and I are here and our temperatures are fluctuating around freezing, we are both, Wisconsin girls at heart. And believe me, it's been all too long since I've been home, and I definitely don't miss it. I know Jen frequents northern Wisconsin a little more often, but I don't miss breaking all those water tubs. So on that, I just want to give all of our speakers a big round of applause again. Thank you guys so much for hanging out and for joining us today and staying on just a little bit longer to answer everybody's questions. It's been great. We'll send everybody information once all of our webinars get posted. I'm going to go stick one more time the evaluation into the chat for me. If you guys will take just 3 minutes of your time and fill it out. But thank you again, everybody, and have a great night.